Good morning, dear students. I welcome you all to this class on PCR, that is polymerase chain reaction. I request you all to view this particular class along with pen and notebook, okay, to make notes. Kindly invest your quality of time here. One more request, if you have not yet subscribed this channel, kindly subscribe this channel to support. Today's lecture talk on PCR module I have prepared for the viewers' benefit with meticulous condition. I know you will utilize this class for your benefit. Before I begin, let's go through today's quote of the day. Every situation is an opportunity. Either you harvest well-being or suffering. It is your choice. I'll repeat. Every situation is an opportunity. Either you harvest well-being or suffering. It is your choice. Now, let's begin today's lecture talk on PCR. Okay. The full form of PCR is polymerase chain reaction. Okay, this polymerase we have taken from the DNA polymerase. Okay, this DNA polymerase, which is responsible for the extension of DNA. What do you mean the extension of DNA? That is nothing but the synthesis of DNA, right? Synthesis of DNA, which is carried out by the DNA polymerase enzyme, or this particular DNA polymerase enzyme, which adds individual nucleotides. What we'll say these nucleotides as DNTPs, right? Deoxynitrogen triphosphate. Individual nucleotides, that is DNTPs, will be added by this DNA polymerase according to the template DNA, right? Now coming to the chain reaction, okay? This chain reaction, that is nothing but the chemical reaction, okay? This is the chemical reaction which proceeds which proceeds in an exponential manner, okay, which proceeds in exponential manner, okay. This PCR can also be known as what? Temperature regulating machine or thermal cycler. Okay, why thermal here? In this particular PCR machine, we are going through this series of temperature. It means the sample of the DNA in the PCR machine will be incubated in a series of temperature, okay, in a repeated cycle manner. Okay, so we'll call this as the thermal cycler. Okay, this PCR is also nicknamed as R. The full form of this PCR can also be known as People's Choice Reaction. Okay, People's Choice Reaction. Okay, hope this is clear to you. Now, what is PCR? If anybody asks what is PCR, what you can say here? What is PCR? This PCR, it's a cell-free technique. You can mention this as this PCR is a cell-free technique. It means in this PCR machine, there won't be any kind of cell. Okay, so you can mention cell-free technique, which is responsible for what? Amplification of DNA. It is responsible for the amplification of DNA. Okay, which amplify the desired DNA molecules into several copies. So the main role of the PCR machine is to amplify the desired DNA molecule according to our desired number of copies. Okay, now let us go through the further about PCR. So in order to study genetic structure and function, we need the sufficient quantities of DNA of interest must be available, right? If we don't have enough amount of the DNA sample, then what happens? Then we cannot perform the some of the experiment and research work, or maybe in the forensic science sample, they cannot compare with the others, right? So we need enough amount of the sample. Earlier, this particular technique was carried out by using microorganisms. This is cell technique, right? microorganisms to generate multiple copies of genes, which is a lengthy and laborious process. Why which is lengthy and laborious process? Because your interested DNA, you need to introduce into the plasmid DNA, and this plasmid DNA you need to introduce into the bacterial host system, right? So after introducing this plasmid DNA into the host system, then we need to grow that in the culture plate, right? So we need to wait for the 24 to 48 hours, then we need to observe, right? So it is a very lengthy and laborious process. So we are depending on a cell-free technique. It means nowadays a cell-free technique is available for the amplification of DNA, that is known as PCR, that is polymerase chain reaction, which is nicknamed as people's choice reaction. Why this is named as people's choice reaction? Because this PCR machine can be seen in almost all the research laboratory. That's why we have mentioned this as people's choice reaction. Okay. Polymerase chain reaction is a what kind of technique? Molecular biology techniques, or a technique used in molecular biology to amplify single copy of a DNA or a few copies of segment of DNA into several orders of magnitude, generating thousands to millions copies of a particular DNA sequence. Okay, and this technique has made it possible to synthesize large quantities of DNA fragment without cloning it, right? Without cloning it in the cell system. Without cloning in the cell system, we are nowadays we are able to produce many number of copies. I hope you are with me. Pay attention, be with me. This technology is much easier and requires much smaller quantity of DNA, that is in nanogram. Okay, whereas cloning DNA means this is the cell system. We need to use the microorganism, bacterial organisms. Okay, this particular cell system for cloning DNA, this needed microgram quantities, but the PCR, the nanogram amount of DNA is found, that is more than enough, that will amplify the uh, desired DNA into many number of copies. Okay, if I'm going very quick, you can note down this page number. Okay, you pause this video and kindly make a note of this PCR. Okay, don't simply sit, kindly make notes and finish it off today only. This particular PCR information for 5 marks and 10 marks, you make your own notes and keep it ready. Okay, this technique was discovered by Carrie B. Mullis in the year 1985, who worked in Citrus Corporation Biotech Company of United States. Okay, PCR was the first major scientific development of the year 1989. It is automated, right, and carried out by a specially designed machine known as PCR. Okay, PCR. This PCR machine, which is a in vitro technique that will amplify the DNA molecule in the in vitro condition. So you can say this is an in vitro DNA amplification technique that allows synthesizing million number of copies of the gene. Okay, gene or DNA of interest from a single copy, or from the nanogram you can get the more number of copies of the DNA molecule. Hope I'm making myself clear to you. PCR can be used to clone a given DNA sequence in vitro without using living cells during the cloning process. Okay. However, this procedure can only be applied when the nucleotide sequence of at least one short DNA segment of each side of the region of interest is known. It means whatever you are going for the PCR machine, no, if this is the DNA strand, okay, if this is the DNA strand which are connected are held together by the presence of hydrogen bonds. Okay, what I said here is that what I said here is that if you want to perform this PCR experiment, you must know the on each side of the region sequence must be known. Okay, if you want to perform this experiment, compulsory you must know the end sequences. So what I have written here, this can only be applied when the nucleotide sequence of at least short DNA segment on each side of the region of interest is known. So in this particular DNA molecule, what kind of DNA sequence is here and what kind of DNA end sequences are there? Okay, we must be knowing that one. Otherwise, we cannot perform. Otherwise, sometimes we won't be knowing it. We can also use the random primers. Okay, we can use the random primers, PRIMERS. Okay, you can use the random primers or you can also use the known primers. Just keep in your mind. I'll come to you later. What is this particular primers? Okay. So hope you understood here. To perform this experiment on either sides of the either ends of the DNA molecule, that should be known. If we do not know, we can also use the random primers. PCR producers make use of synthetic oligonucleotides, okay, which are complementary to the known sequences. Okay, so what do you mean by here? This synthetic oligonucleotides, which are complementary known sequences, that is nothing but the primers. Okay, primers are the synthetic, that is artificially prepared oligonucleotides, few nucleotides, which are complementary to the known sequences. Whatever the DNA molecule will be there, no, what we treat the primers here, these primers are single-stranded synthetic oligonucleotide, which are complementary to these particular sequences. Polymerase chain reaction may also be defined as a technique in which repeated enzymatic thermal cycles of the DNA synthesis can be carried out, okay, can be carried out to produce large number of specific DNA sequences. Okay, this technique is so precise and powerful that even single sequence can be amplified into a detectable amount. Okay, then now let us go through the principle. The polymerase chain reaction is a powerful and widely applied technique or used technique that has greatly advanced our ability to analyze genes. Okay, chromosomal DNA present in cells contains thousands of genes. This makes it difficult to isolate and analyze any individual genes, right? Whereas PCR allows specific DNA sequence, usually corresponding to genes or parts of genes. Okay, the only requirement is that some of the DNA sequence at either end of the region to be copied is known. What are the DNA molecule be there? Either ends of the DNA molecule that should be known. If we know that particular either ends of the sequences, then we can go for the performing the PCR. This is the ends. Okay, or if your DNA is here but your no sequences are here, so this particular sequences must be known to prepare primers. Why we should know the two ends of the DNA sequences to prepare primers? Just keep in your mind. Next slides you'll be able to make out. DNA corresponding to a sequence of interest is copied or amplified by PCR more than a million fold and becomes the predominant DNA molecule in the reaction. Okay, it is sufficient where sufficient DNA is obtained for detailed analysis and manipulation of the amplified gene in research area. Okay. Now let us go through the mechanism of PCR. In order to understand PCR, it is necessary to be familiar with the mechanism of DNA replication. Okay, what is happening in DNA replication? Same concept we have done in the PCR. It means this DNA replication within the cell system, which is the in vivo condition, the same mechanism we are applying in the PCR machine in the in vitro condition. Okay, the DNA replication involves polymerization of nucleotides, right? Using template DNA. What do you mean by template DNA? Template DNA is the single stranded DNA. Okay. Strand with the help of oligonucleotides, which acts as primer strand to which further nucleotides can be added using the DNA polymerase enzyme. Okay. So in the replication, what happens if this is the DNA molecule? They need the primer, right? Once the primers are ready, right, with the DNA polymerase enzyme, then this ends will be synthesized by the DNA polymerase enzyme, right? So these primers are known as, okay, these primers can also be known as oligonucleotides. Okay, these are the primers over here. Now let us go through the components which are required to perform this PCR experiment. Okay, DNA is amplified by PCR in an enzyme, okay, reaction which undergoes multiple incubations of at different temperature, because here the sample of PCR which undergoes multiple incubations, right, at different temperature. That's why this PCR can also be known as thermal cycler. Each PCR has 4 key components, okay, as follows. First key component is the template DNA. Okay, first there will be a double stranded DNA molecule which are held together by the hydrogen bonds. Okay, once these hydrogen bonds are broken down, you are going to get this in the form of single stranded DNA, right? Once the hydrogen bonds are broken down, single strand. This single stranded DNA are known as what? Template DNA. Okay. Once this template DNA is ready, we need oligonucleotide primers. Okay. What are primers means? Primers usually somebody will say 20 nucleotides, somebody will say 30 nucleotides, somebody will say 50 nucleotides. So you can say primers are the single stranded known nucleotides which are of 20 to 30 nucleotides. And these oligonucleotide primers are complementary to the ends of the target DNA. Okay, and they help in initiating extension or synthesis of the DNA. Okay, so let us go through here. In the absence of oligonucleotide primers, DNA polymerase cannot synthesize the DNA. Okay, so template DNA we have. To this template DNA, primers goes and attach. Okay, when the primers will go and attach, when they have the complementary nitrogenous bases that goes attached to the ends of the DNA molecule. Okay, these primers are the known sequences. Hope you are getting here. So these are the primers. Okay, primers are single stranded 20 to 30 nucleotides long. And what is the function of these oligonucleotide primers? They help in initiating extension or synthesis of the DNA molecule in the presence of DNA polymerase enzyme. So in ethylene, so earlier DNA we had this hydrogen bonds are broken down, then we got the single stranded DNA, which is known as the template strand. To this template strand, oligonucleotide primers that will goes and binds on either ends of the DNA fragment. Then DNA polymerase comes, our hero of this PCR. Okay, this DNA polymerase enzyme that goes and sit here wherever they have the initiation of double stranded region. Okay, always DNA polymerase can synthesize or initiate that particular extension only in the double stranded extension. Okay, for example, if the template DNA is here, if there is no primer attachment on the ends of the DNA molecule, your DNA polymerase cannot synthesize only on the template DNA. Okay, they need the double stranded initiation. Okay, once the primer attached, after attachment of the primer, then this DNA polymerase goes and sits and starts synthesizing the synthesizing what? Individual nucleotides according to the template DNA. According to this is template DNA, right? According to template DNA, that will keep on adding the individual nucleotides. Okay, this is the very simple mechanism of PCR. So, what are the major requirements? Template DNA, oligonucleotide primers, then DNA polymerase. So, in the component, what else require? We need DNTPs. What do you mean DNTPs? Deoxynucleotide triphosphate. This is individual nucleotides you need to provide, because when you are providing template DNA primers and DNA polymerase enzyme, you need to provide in the reaction tube, because this is happening in the in vitro condition. In the in vitro condition, you need to provide everything, right? So you need to provide the individual DNTP, that is deoxynucleotide triphosphate. You need to provide in the reaction mixture. Many number of, how many number of copies? That many number of individual deoxynucleotides you need to provide. Okay, then most important one, reaction buffer. So all these components will be suspended in the reaction buffer. So this is about the components of the PCR. Now we can see here in this particular reaction mixture, we can see the presence of DNA molecule. Okay, then you can see the individual nucleotides. Then you need the polymerase enzyme. What kind of polymerase enzyme? This is the Taq DNA polymerase enzyme. I'll come to this later. Just keep in your mind. Okay, when you're going through your higher studies, usually in entrance examination, also they'll ask this PCR. Even if you're writing or attempting IIT JAM examination or any other entrance examination, usually you'll get the questions from PCR. So pay attention here. So what kind of enzyme will be using in this PCR? I said only polymerase word, right? Now I mentioned here Taq polymerase, or you can also say Taq DNA polymerase. So keep in your mind, this thermal cycler or extension of synthesis will be carried out in the presence of Taq DNA polymerase. Okay, so you can say Taq DNA polymerase, and this is the single strand primer. So these are the requirements to perform polymerase chain reaction. Okay, now let us understand what is this template DNA. Template DNA containing target DNA sequence to be amplified. Okay, the template DNA is a complex mixture of many different sequences. Okay, any DNA molecule that can be used as target sequences. RNA can also be used for PCR by first making this particular single strand RNA molecule into cDNA by using which enzyme? Reverse transcriptase enzyme, right? By using reverse transcriptase enzyme, we can convert this mRNA into DNA molecule, that is cDNA molecule, by using the enzyme reverse transcriptase. Whenever we write anywhere we have cDNA sample, you must understand this cDNA we have obtained from the mRNA molecule. Okay, so what is template DNA here? Template DNA contains the target DNA. Also, we can say template DNA are single-stranded DNA. Okay, template DNA is a single-stranded segment or single-stranded DNA molecule. Now, let us go through here oligonucleotide primers, whereas oligo means few nucleotides. Few nucleotides of primers can be seen, few nucleotides of around 20 to 30. Each PCR is required a pair of oligonucleotide primers. Okay, what are primers? Somewhere you'll get this question even in Viva or maybe in MCQ also. Primers are short single-stranded DNA molecules, typically 20 bases. Some of the books they'll also mention 30. So I can write 20 to 30 bases obtained by chemical synthesis. In the in vitro condition, these primers are synthesized. Okay, you can also mention these are short single-stranded non-DNA molecules. You can also go for random oligonucleotides here, but you can mention here these are non-DNA molecules. We'll be knowing what kind of primers we are prepared. Primer sequences are chosen so that they bind by complementary base pairing to the amplified DNA or amplifying DNA. If this is the DNA template strand, okay, we are choosing the primers in such a way that that primer should go and bind to the ends of the DNA fragment. Once they bind to these ends of the DNA fragment, you can see the double stranded region. Now your DNA polymerase enzyme will be ready. Okay. this DNA polymerase enzyme cannot synthesize on single standard DNA. This where this will go. This can't bind to this template DNA and they cannot initiate. They need the DNA polymerase for the initiation or extension of synthesis compulsory. They need the double standard initiation region. Once they get this kind of double standard initiation region, then they'll start synthesizing the individual DNA molecule. Okay, now let us understand here. Okay, before that, let me talk about more about primers here. So when we consider this particular primers, I told these are the single standard DNA and they need and this particular single standard DNA which are needed by the DNA polymerase enzyme for the initiation of the DNA molecule or synthesis of the DNA molecule or extension of the DNA molecule. One more thing you should keep in your mind, we are using here in oligonucleotide primers two types of primers. Okay, do you have any answer? What are those two types of primers? That is forward primer. Okay, other one is the reverse primer. Okay, in this particular oligonucleotide primers, we are using two types of primers. Okay, that is forward primer and the reverse primer. For example, this will be, just imagine this is 5' to 3' direction. Other strand will be 3' to 5' direction. Okay, so here at this particular primer region, this is 5' to 3'. At this particular region, the primers goes and binds, right? If this is the 3', this will be your 5' and this will be your 3'. If this is the 3', this will be your 5' and this will be your 3'. Synthesis always begins in shape from 5' to 3'. So this will be known as forward primer. Okay, this is the forward primer, and here from this side to this side, synthesis will be happening in the presence of the polymerase enzyme. So this guy, the primer which is attached here, right, to the this from this particular region, this will be known as reverse primer. This is forward primer here, okay, below one, and this will be known as reverse primer. Okay, this is the forward and this is the reverse primer. So this is about the oligonucleotide primers. Now let us understand the next mechanism, that is annealing. Okay, annealing of the primer to the template acts as initiator for DNA synthesis. Annealing means that is nothing but attachment or sealing. For example, we have a double stranded DNA molecule. In this double stranded DNA molecule, here the hydrogen bonds will be broken down. Once the hydrogen bonds are broken down, you will get the template DNA. To this template DNA, the single stranded DNA, that is single stranded primers which goes and attach, that attaching or sealing of this particular primers, okay, to the template is known as annealing. Annealing of the primer, attaching this is the primer, right? This is the primer and this is the template, right? This is the template region and this is the primer, short stretch of DNA molecule, right? This primers are single stranded, right? Single ಹೋಗ್ಬಿಟ್ಟು template attach ಆಗಿದೆ. Single stranded DNA, right? So in the annealing step, what happens? Annealing or sealing or joining of the primer. Joining of the primer to the template, which acts as the initiator for synthesis. Okay. For example, if this is the 5' to 3', this will become the forward primer. Okay. Here DNA polymerase goes and binds. Okay. And once that gets the double strand initiation, then that will start synthesizing the individual nucleotides according to the template DNA. Here also the tag DNA polymerase goes and binds and synthesize the individual nucleotide according to the template DNA. Okay. Hope you understand. If there is only a template, there is no primer attachment, they don't have any kind of affinity, then the tag DNA polymerase cannot sit anywhere, because tag DNA polymerase, they require compulsory double stranded initiation region. Okay. Oligonucleotides are small stretch of desired fragment which acts as a primer to synthesis DNA. Okay, that is primers are initiators of PCR which are obtained by chemical synthesis. That is in the in vitro condition. Now let us understand in detail here about the DNA polymerase. Okay, a major improvement in PCR amplification of DNA came with the discovery of heat-stable DNA polymerase. Heat-stable means this particular DNA polymerase can withstand to high temperature. Okay, so that's why you can mention this as heat-stable DNA polymerase. The most commonly used enzyme is Taq DNA polymerase. Okay, this Taq DNA polymerase, which is obtained from Thermus aquaticus. Okay, Thermus aquaticus bacterial organism or bacterium, which is of archaebacterium growing in hot springs. From this particular bacterium, Thermus aquaticus, this Taq DNA polymerase enzyme was isolated. Okay, this enzyme can withstand temperature above 95 degrees Celsius. Okay, and this enzyme which also have exonuclease activity. Okay, this enzyme which also exhibit exonuclease activity. Exonuclease. Okay, exonuclease activity. And this enzyme responsible for synthesis of the DNA molecule according to the template DNA or this extend the primer, okay, based on the template. For example, this is the template DNA. Primers are attached over here. Okay, hydrogen bonds which are formed here. Once they get the double-strand initiation, Taq DNA polymerase goes and binds there. And this Taq DNA polymerase keep on adding the individual nucleotides according to the template DNA. Okay, and as and when this Taq DNA polymerase moves, which is going to add the individual nucleotide. Same thing here also it will be happening. Individual nucleotide will be added by the DNA polymerase enzyme as and when moves. Okay, further. The polymerase is called Taq polymerase after the genus and species name of bacterium, that is Thermus aquaticus, from which it was isolated. Okay, hope you understood here. This polymerase enzyme responsible for extension of DNA, okay, then synthesis of DNA, right? Or this particular DNA polymerase enzyme which adds the individual nucleotide according to the template DNA. Okay. The polymerase enzymes acts best at 74 degree Celsius. It means this is the optimum temperature for the Taq DNA polymerase. In some of the books, they'll also mention 72 degree. Okay. You can write 72 degree or 74 degree Celsius. And also remaining active during heat denaturation step. Okay. Why I mentioned here they remain active during the heat denaturation step, because in this particular PCR machine, the samples will be incubated up to 90 degree Celsius. It means, as I told, the Taq DNA polymerase can withstand up to 95 to 97 degree Celsius. Whereas in this case of denaturation process, the instrument will be increased temperature to 90 degree Celsius or more than 90 degrees Celsius in each cycle of amplification. That time, this particular Taq DNA polymerase can withstand to high temperature. Okay, that's why it is mentioned as this Taq DNA polymerase thermal stable. Okay, that's why this particular PCR machine can also be known as thermal cycler. Okay. Now let us understand the role of the Taq DNA polymerase in PCR is to copy DNA molecules. Okay. The enzyme binds to double-stranded DNA and synthesize a new strand complementary to original strand. So what do you mean by this? This is the template DNA. Okay, right. This is the template DNA. Here the primers goes and binds over there on the three ends of the DNA. fragment, then the DNA polymerase enzyme goes and sits over there, right? The enzyme binds to double-stranded DNA initiation and synthesis new strand. Okay, new strand will be synthesized here on both the side at a time. DNA polymerase will start synthesizing based on what? Which are complementary to the original strand. Based on complementary originals, based on this original strand, complementary nitrogen bases will be synthesized. DNA polymerase requires short region of double strand DNA to get started. Beginning, if they don't have this short region of DNA along with primer, they cannot initiate. They need compulsive double strand initiation region. In PCR, this is provided by oligonucleotide primers. What is provided? The initiation double stranded region is provided by the primers, which creates short double stranded regions by binding on either sides of DNA strands to be amplified. Okay. In this way, the primers direct the DNA polymerase to copy only the target sequence. Okay. So that is about the tag DNA polymerase. Now let us understand here deoxynucleotide triphosphate. So these molecules correspond to the four bases which are present in DNA, that is adenine, guanine, thymine, and cytosine. As this PCR which is meant for the DNA amplification. For DNA amplification, we are using which enzyme? DNA polymerase enzyme. Okay. Whereas in conversion of mRNA to cDNA, we'll be using reverse transcriptase enzyme. Okay. Each PCR requires 4 DNTPs. Okay. So what are those? Deoxyadenosine triphosphate, deoxycytosine triphosphate, deoxythymine triphosphate, deoxyguanine triphosphate, which are used by the tag DNA polymerase as building blocks to synthesize new DNA molecule. Okay. PCR allows the amplification of target DNA sequences through repeated cycles of DNA synthesis. That's why it is said thermal cycle. Along with series of temperature, there are so many cycles we are going to repeat. That's why it is said cycler. Okay. Each molecule of target DNA synthesized acts as a template for the synthesis of new target molecules in the next cycle. As a result, the amount of target DNA increases with each cycle by using the reaction components. It means what I mentioned here, each molecule of target DNA synthesized acts as template. It means, for example, here I have a double-stranded DNA molecule which are held together by the hydrogen bonds. Okay. This here in this case of PCR, what we'll do, we'll break this hydrogen bonds. Once we break the hydrogen bonds, the two strands will get separated. And this will be known as what? Template DNA. Okay. And to this template DNA in the PCR reaction mixture, the primers goes and attach. Okay. Primers goes and attach. That is nothing but annealing, right? Annealing of the primer. Once annealing of the primer done, we already subjected DNA polymerase enzyme, tag DNA, right? This tag DNA which goes and binds to the double stranded initiation region of the DNA molecule, right? This double strand region that goes and binds. Okay. Once that goes and binds to that region, this DNA polymerase enzyme, it keep on adding, okay? This DNA polymerase enzyme moves, okay, like an earthworm that moves, and this is going to add the individual nucleotide at a time. This will add the individual nucleotides according to the template DNA, right? Here also you can see here. So this DNA which is going to add the individual nucleotides according to the template DNA, okay? And here also this will add the individual nucleotides according to the template DNA as and when this particular tag DNA polymerase moves in this direction, here this in direction. Once after the synthesis, next what happens? This particular tag DNA polymerase will detach from that particular DNA molecule. be with me. Okay. In this case, what happens? This is the original DNA molecule, right? And this is the primer. Whereas in this case, this is the template DNA molecule, original strand, and hydrogen bonds already there, which are held together, right? So this is held together. So earlier we had actually through template two strands of DNA. After the amplification, then the stacked DNA polymerase enzyme will detach. Okay, this will detach. So now we have from the two strands of DNA, this is template and this is template. We had only the two strands. From the two strands in this reaction mixture, we got the new strand. Next, what happens here? This particular DNA molecule, okay, both strand will get separate and again they'll become template DNA strand. And they become template DNA strand. It means each and whenever they synthesize the new strands, no, those new strands will become the template. For example, this strand is this one, okay? The hydrogen same repeats. Hydrogen bonds will broken down. Once the hydrogen bonds are broken down, the primers don't attach, okay? This is this strand. Okay, these hydrogen bonds, here these hydrogen bonds will broken down and they separate like this is the single strand original DNA and newly synthesized. The primer one will be there. This is newly synthesized. Whereas in this case also, this original DNA, this is the original DNA, this will get separate and this particular newly synthesized DNA can be observed here with the separation. Okay, here you can see this is the newly synthesized. And again, here also same thing, primers going attach, here also primers going attach, and here also primers going attach, here also primers goes and attach. Tag DNA polymerase goes there. Okay, here also tag DNA polymerase, here tag DNA polymerase, here tag DNA polymerase. Then they'll keep on adding the individual nucleotides. Here also they keep on adding the individual nucleotides. Okay, then here also they'll keep on adding the individual nucleotides, and here also they'll keep on adding the individual nucleotides. So how many strands now we have? 1, 2, 3, 4, 5, 6, 7, 8. Okay, from 4 strands we got here the further another 4 and they become 8 strands. Earlier we had 2 strands, then here 4 strands, then 8 strands. Okay, same cycle repeats. Then this particular DNA molecule strand, again this will separate and they become single stranded. Again each time the newly synthesized and template DNA strand which becomes the template strand for the further cycle. Okay, hope you understood here. Now let us understand each cycle of DNA synthesis involves 3 stages: denaturation, primer annealing, and polymerase extension. Okay, so now let us go through the other total overall requirements of the PCR machine. The first one is that is target DNA. We need the target DNA to perform this particular experiment, right? What is target DNA here? The target DNA which contains the sequence to be amplified. Okay, target DNA, that is nothing but sequence to be amplified, that will be known as the target DNA. So to perform this experiment we need compulsory target DNA. Then we need 2 oligonucleotide primers. What kind of 2 oligonucleotide primers? We need the reverse primer and we need the forward primers, both we need, right? Then these, what is the job of the primers? Primers gives which goes and binds to the 3' hydroxyl of both end of strands. Okay, for example, if this is your DNA molecule, okay, if this is another DNA molecule, what is the job of the primers? The primers which goes and binds to the 3' end of the DNA molecule. For example, let me write here, this is 5' and this is 3'. If this is 5', this will be your 3' and this will be your 5' because anti-parallel nature, right? The primers goes and attach. So primers go attached to the 3' end. Okay, here one 3' end and here that will go 3' end. When this is the 3', what this primer will become? This will be 5' and this will be 3'. If this is the 3 primer, opposite end will be, this will be 5 prime end and this will be 3 prime end. So what is the job of the primers? Two oligonucleotide primers goes and binds to 3 prime hydroxyl ends of both strands. So which is reverse primer and forward primer? We can say this as the forward primer and this as the reverse primer. Okay. Then most important one is DNTPs, deoxynucleotide triphosphate. That is, this DNTPs are like building blocks or building units of this particular DNA synthesis. You need to provide many number of DNTPs like adenine, guanine, cytosine, thymine you need to provide. Next most important component is thermostable DNA polymerase, that is TAC DNA polymerase, enzyme that catalyze the reaction or enzyme that synthesize the DNA molecule or extend the DNA molecule or add the individual nucleotides. Then we require magnesium ions. This magnesium ions, which is going to act like a cofactor of the polymerase enzyme. Okay, all this need to provide in the reaction mixture. Whereas in the in vivo condition, what happens? Everything that will be in the cell system, cell will provide. Whereas when you are doing in this in vitro condition, no, that time you need to provide everything. So we need to provide the magnesium ions as a cofactor to the polymerase enzyme. So next one is the most important one, buffer solution. This buffer solution we are using in this PCR machine tube to main with this buffer which maintains the pH and ionic strength of the reaction solution, which are suitable for the activity of the enzyme. So in this particular reaction mixture, all this will be suspended. Okay, let us imagine, you can see here the reaction mixture. Okay, in this reaction mixture, you are going to suspend what? You are going to suspend the target DNA. Okay, the first one, target DNA are going to suspend. Then oligonucleotides are primers. Okay, you are not going to suspend target DNA only one. There will be many copies. Okay, target DNA are going to suspend, then primers. Then you need to provide in this particular reaction mixture DNTPs. Okay, DNTPs, stacked DNA polymerase, and magnesium ions you are going to provide. Okay. Kindly make a note, simply don't watch, otherwise you'll forget. Now let us understand the three mechanisms of PCR. The first one is denaturation. Okay, here we are doing heat denaturation. Heat denaturation, that is nothing but the melting of target DNA. For example, in the reaction mixture, you are going to provide the DNA molecule always in the single standard condition. Okay, in the reaction mixture, the double stranded DNA molecule will become single stranded DNA after denaturation process. That is nothing but the heat denaturation or melting of target DNA. When we subject this particular DNA molecule to the temperature around 95 or 92 degrees Celsius, that time hydrogen bonds will be broken down and the DNA strand hydrogen bonds breaks and you'll get the single stranded DNA, and this is known as the template, right? So what is this denaturation? The mixture is heated up to 94 degrees Celsius, or some of the books they'll mention 92 or 94 or 95, any one you write. Okay, at that temperature, the hydrogen bond that holds together the two strands of the DNA molecules are broken, causing the molecule to denature. So this is nothing but the denature. When we expose this DNA molecule to 94 degrees Celsius or 92 or 95 degrees Celsius, the hydrogen bonds will be broken down. Once the hydrogen bonds are broken down, you will get the single stranded DNA like this. Okay. Now coming to the primer annealing. You know the definition of the primer. Now let us understand what is this primer annealing. Primer annealing is the mixture is cooled down to or lower to 50 to 60 degrees Celsius. Okay, a temperature that allows binding of the primers to the single stranded DNA without permitting the double helix to reform between the template strands. Okay, it means earlier we took the double stranded DNA molecule, right? We took the double stranded DNA molecule. This double stranded DNA molecule was subjected to the denaturation process. What temperature? What temperature? 94-degree temperature. Okay. When we increase the temperature in the PCR machine to 94 degrees temperature, what happens here? This double strand DNA molecule hydrogen bonds will be broken down and they separate like this and they become template DNA. Okay, and this is the template DNA. Once we get this kind of two template DNA, next what happens in this particular instrument, we are going to lower the temperature or we are going to reduce the temperature to what's temperature 50 to 60 degree, okay, Celsius. 50 to 60 degrees Celsius. When we reduce that time, what happens? This is the optimum temperature for the annealing or for the attachment, okay, or sealing of the primer DNA, which happens with the free ends of the template DNA. I told you already that always this primers will goes and attach to the 3' ends of the template DNA molecule. Okay, let me repeat here. Primer annealing. Annealing means joining or just a binding. So this mixture is cooled. This mixture means that PCR tube mixture is cooled down to at lower to 50 to 60 degrees Celsius, a temperature that allows binding of the primers to the single stranded DNA without permitting the double stranded helix to reform between the template strands. Okay, that's the 50 to 60 degrees Celsius. Only these primers which goes and attached by avoiding the binding of two template DNA strands. And this process is known as annealing. Somewhere in the question paper, they want this one for two marks. Okay, keep in your mind. You can put one star mark. Primers are the initiation of PCR that anneal to the DNA molecules at specific position. Why this is the initiation of PCR? Because we have a tag DNA polymerase enzyme for the synthesis of the DNA molecule, right? Tag DNA polymerase enzyme. This any polymerase enzyme cannot synthesize the another strand only on the template DNA strand. They need compulsory double-stranded initiation. Okay. Once they get the double strand initiation, then they start the synthesis of the another complementary strand. Hope you are getting here. So this is all about the primer annealing. Okay. Now let us go through the extension or elongation. Okay. Or polymerase extension or synthesis. Okay. So the third step is extension or polymerase synthesis. So this stage is carried out at a temperature at which the DNA polymerase is most active. And this is the optimum temperature. What is that? The temperature is raised to 72 degrees Celsius. Okay. And this is the optimum working temperature for the tag DNA polymerase that is present in the reaction mixture. All this is happening in the, where this is happening, it's in the reaction mixture. Okay. First, we have put the double stranded DNA molecule. Then we have put the individual single stranded primers, right? Now we have put the tag DNA polymerase enzyme. So first, what has happened? First, we have took the double stranded DNA molecule here. This double stranded DNA molecule reaction mixture was increased to the temperature that is 94 degrees Celsius. That time what happened? The double stranded DNA molecules get separate and they become template, right? Okay, and they become template. To avoid these two DNA molecules of rebinding, the instrument is reduced to the temperature that is 50 to 60 degree Celsius. At this particular temperature, what happens to this double stranded DNA molecule? Whatever we have subjected here, the primers know that primers which goes and attach to the 3' hydroxyl group of the both the ends of both the strands of the DNA molecule or template DNA molecule. Okay, once they bind, they'll get the they got the double strand initiation. That time what happens? This tag in this case, once 52 degrees Celsius is happened, again in the instrument, the tag DNA polymerase enzyme optimum temperature is that is 72 degree Celsius. The instrument is again increased to the temperature 72 degree Celsius. At that time, what happens? What are the primers which are attached here? No, to the double strand initiation region, what happens? When we increase the temperature to 72 degrees Celsius, the tagged DNA polymerase goes and binds to the double strand initiation region and they start synthesizing the complementary DNA molecule. At what temperature? 72 degrees Celsius. Okay. You can see here series of temperature. Okay. That's why it is mentioned as thermal. See, first increased this PCR machine to 94 degrees Celsius, then 50 degrees Celsius, then 72 degrees Celsius. Okay. This is one particular cycle. At 72 degrees Celsius, you can see this particular synthesis of the double stranded DNA molecule can be seen. Okay, the same cycle repeats. This double stranded DNA molecule, again that will be going through the process of separation of DNA molecule, which is known as what? Denaturation. So denaturation process will be happening at 94 degrees Celsius. After denaturation, 50 to 60 degrees Celsius, we are going to reduce, once we reduce the temperature, we'll say this technique as annealing. After the annealing of the primer, we have increased this PCR mixture machine temperature 72 degree Celsius. At this 72 degrees Celsius, that DNA polymerase goes and attaches to the double stranded initiation region. Then they start synthesizing the individual nucleotide according to the template DNA. Once this tagged DNA polymerase synthesis, that will detach. Okay, and again this cycle repeats. The cycle go on repeating, denaturation, annealing, extension, denaturation, annealing, extension. Okay, like that, that will be happening. That's why it is known as repeated thermal cycler. Here, tagged DNA polymerase attaches to the end of each primer and synthesizes new strands of DNA, right, which are complementary to the template DNA. Now, we have 4 strands of DNA instead of 2. From 2, we got the 4 strands, 1, 2, 3, and then 4. Newly synthesized are the 2. That's why instead of 2. This completes one cycle of PCR, denaturation, annealing, and extension. Okay, extension at 72 degrees Celsius. This is one cycle. The same cycle repeats again and again. That's why it is also known as repeated thermal cycle. Okay, the temperature is increased back to 94 degrees Celsius. Then the double stranded DNA molecules, each of which consists of strands of the original DNA and one set of new strands of DNA, again denature into single strand DNA. Okay, hope you're able to make a note of this one. This begins a second cycle of denaturation, annealing, and synthesis. At each end of cycle, there are 8 strands obtained. Okay, for example, this is the double strand DNA in the beginning, right? In the beginning, this is the double stranded DNA molecule. This double stranded DNA, further temperature was increased to 92 degrees Celsius or 94 degrees Celsius for the denaturation process. When we increase the temperature to 94 degrees Celsius, hydrogen bonds will be broken down and that process is known as denaturation. Once denaturation happens, then this template can be seen. To this template, in the PCR mixture, we are going to increase the temperature, reduce the temperature or lower the temperature to 50 to 60 degrees Celsius. That time what happens? What happens? The primers goes and attach to the 3' hydroxyl region of the template DNA strand. Okay, and this will be known as primer annealing. Once the primer annealing is done, then next we are going to increase the temperature to how much? We're going through the 72 degrees Celsius. 72 degree temperature is the optimum temperature or working temperature for your tag DNA polymerase enzyme. So we'll say this as the extension. Okay. So in this case, what happens? Whatever the template DNA will be there, primers which are attached over there, right? Primers which are attached over there. In this case, what happens? The tag DNA polymerase goes and binds to the double strand initiation region on both the strand. Okay, and that at that 72 degrees Celsius, it will go on adding individual nucleotides according to the template DNA molecule. Okay, according to template DNA, that goes on go on adding. That time, how many strands you got? Earlier you had only the 2 strands. Now we have 4 strands. Okay, the same cycle repeats. The temperature is again, see here, here this is 94 degree. 50 to 60 degrees Celsius, then here 72 degree, right? This is one particular cycle, okay? This is one cycle, okay? The same cycle repeat, the temperature is increased, okay? The temperature is increased back to 94 degrees Celsius. So here the temperature was 72 and here the temperature was 50 to 60, 94. Here this particular four strands where they know that were also increased, subjected to again denaturation process. Okay, this strand further it will go for the denaturation, right? And this strand also further go for the denaturation process. Okay, the strand will go through the denaturation process, they'll split, and further they'll go through primer annealing, and then further they'll go through the extension. Okay, here also same thing, denaturation happens, then primer annealing happens, then extension happens, then from this particular 1, 2, 3, 4 strand, you are going to get the total 8 strands you are going to get. It means how these two strands are going to separate like this. These two strands will separate like this. Again, primers goes and attached, individual nucleotides are synthesized. Here also primers attached, individual nucleotides synthesized. And here also primers attached, individual nucleotides synthesized. And here also primer attached, individual nucleotides synthesized, primers attached, and individual nucleotides synthesized. So total you get here the 8 strands of DNA segment can be seen. Okay, hope you are able to understand this particular concept here. By repeating this cycle 30 times, the double standard molecule is converted into 2 billion new double standard molecules. Okay, annealing. What is the definition of annealing? Please make a note of this one. Some of the question paper they've asked. The pairing of complementary single strands of DNA to form double helix in PCR. Okay, so that is known as the annealing. What is primer here? Primers are a short sequence of oligonucleotides that hybridize with the template DNA strand and provides initiation for the DNA synthesis is known as a primer. Okay, you can also mention here as the primers are artificially synthesized single stranded DNA molecule. Okay, so now let us go through the steps of PCR. A PCR cycle consists of 3 steps. Okay, the step 1, what is the denaturation, primer annealing, and elongation or extension. Okay, these are the 3 steps: denaturation, primer annealing, elongation or extension by the thermostable DNA polymerase. So this is the brief view of the PCR. If you understand this particular one slide, no, you have understood the entire PCR mechanism. What do you mean by denaturation? Denaturation, that is nothing but the breaking of the hydrogen bonds. Hydrogen bonds will be broken down, right? In the when this denaturation will be happening, this reaction mixture when heated to 94 degrees Celsius for 1 minute, that time what happens? The double stranded DNA, which will become into a single stranded DNA. Okay, the temperature denatures double stranded DNA into 2 single strands by breaking hydrogen bonds between them. So, and they become template DNA. Okay, so to become template DNA, denaturation is very important at 94 degree Celsius. Then step 2, primer annealing. Okay, primers are single stranded non oligonucleotides. Annealing is the attachment or complementary base pairing over here. So when we have already the template DNA is here, then what happens? We have increased the temperature 94 degrees Celsius here. The PCR mixture or reaction mixture is cooled to 50 to 60 degrees Celsius or 55 degrees Celsius for 1 minute. Okay, so in this case, within 1 minute, denaturation will be happening. Okay, denaturation will be happening within 1 minute. Another 1 minute, when we reduce the temperature to 50 to 60, for example, 55 degrees Celsius, primer goes and binds to the 3' ends of the target DNA at both strands. Okay, that is known as primer annealing. Okay, on both these strands. For example, if this is the 5' and if the 3' opposite 5', always 3', and this will be 5'. So what I told, primers binds to the 3' end of the target DNA. Here one primers which goes and binds here. Okay, and here one primer which goes and binds. Okay, what kind, we have two types of primer, no? This by primer which is moving like this, further 5 prime to 3 prime, this will be known as forward uh, primer, right? Whereas in this case, this primer will be known as the reverse primer. Okay. Then let us go to step 3. Elongation by thermostable DNA polymerase at 72 degrees Celsius. This 72 degrees Celsius is the optimum working temperature for the tagged DNA polymerase. And this here, the extension is required around 2 minutes. Okay. Here in this case, what happens? Whatever here, the template DNA primers attached over there, 55 degrees Celsius. When temperature increases again to 72 degrees Celsius, here what happens? The tagged DNA polymerase goes and binds at 72 degrees Celsius at this particular position. Okay, that goes and binds. Then what happens? This tagged DNA polymerase enzyme, which is going for adding the, which will go on adding the individual nucleotides according to what? According to the complementary DNA molecule. According to this complementary DNA, according to this template DNA, the tag DNA polymerase will keep on adding the individual DNT piece. Okay. So here also same thing, individual nucleotides will be added by the tag DNA polymerase, and this tag DNA polymerase moves like an earth form. At a time that will add only one nucleotide. Okay, duration complete cycle will be 4 to 5 minutes. Each cycle, the number of DNA gets doubled. That is from the 2 to 4, 4 to 8, 8 to 16. Like that exponential manner can be seen. Okay, exponential manners can be seen with particular PCR machine. The same cycle repeats. Earlier we had only the two strands of DNA molecule. This strand again, that will be going to the denaturation, annealing and extension process. Again, this strand also will repeat the cycle, the same thing, denaturation, annealing and the extension. Every time what happens? The step 3, whatever the step 3 we have now, this step 3 will go through again step 1 and 2 and then 3. Again, step 3 will go through the step 1, step 2 and then step 3. Hope you're able to understand this particular steps. You can take screenshot and can make a note. Here you can see in the PCR machine, as a sample, you can use the plasmid DNA or genomic DNA, or you can also use the PCR products if already prepared. Then you can also use the annealed oligonucleotides, or you can also use the cDNA, which are extracted from the RNA sample. Okay, we are doing this particular cDNA sample in the RT-PCR. Okay, reverse transcriptase PCR we are using. We have total the 3 types of PCR, that is conventional PCR, real-time PCR, and RT-PCR. Okay. Right now, whatever the coronavirus is there, that coronavirus is RNA molecule. That corona molecule, coronavirus, that RNA molecule made up genetic material is RNA molecule. No, that RNA molecule will be subjected to the RT-PCR, where we are converting that RNA molecule into cDNA molecule. Okay, cDNA synthesis will be done. It means to detect the presence of coronavirus, lab technician people are taking the swab from the nasal and also from the throat. No? Once they take, they'll transfer into the tube, and that tube will be further subjected to the RT-PCR. Okay, we'll discuss any other day what is this RT-PCR. Okay. So here you can see here this is the strand of the DNA, which is already denatured. Once the denaturation occurs, we are going to put the primers here. This is the forward primer, which begin from 5 prime to 3 prime here, right? This is the 5 prime to 3 prime, moving this the reverse primer. Okay, this is the denaturation process. Once the denaturation happens, jagged DNA polymerase goes and binds, and further amplification can be seen here. First cycle, from the 2 strands, you are going to get the 4 copies. Then second cycle, from the 3 cycles, you are going to get the 8 copies. Like that, amplification can be seen. So you can make a note of this one. First cycle, you'll get 4 copies. Second cycle, you'll get 8 copies. Third cycle, 16 copies. Fourth cycle, 32 copies. And 30th cycle, if you perform, you're going to get the 2 billion copies. Okay, so this is your single stranded DNA molecule, which is known as template DNA. To this, your tag DNA polymerase goes and binds over there, okay, and starts synthesizing the it is the new strands which are synthesized. While synthesizing new strand, complementary, right, complementary individual DNTPs will be added over here, okay. Whatever the kind of complementary nitrogen bases will be there, respective DNTPs will be added by the tag DNA polymerase enzyme. Hope you understood the entire concept. Let me repeat again here. In the PCR mixture, you are going to use primer and tag DNA polymerase and the target DNA as that is DNA, then individual nucleotide that is DNTPase. Okay, so this is the PCR machine. You can see here this will be similar to like colorimeter. Okay, so you're going to place it here the PCR cuvette here. Okay, PCR mixture going to put. Okay, we need target DNA sample, then primers and nucleotides, then tag DNA polymerase, reaction mixture, and this is the PCR tube. In this PCR tube, we are going to put all this and we are going to subject to the PCR machine. So what is PCR? Well, we can say it is a thermal cycler or molecular Xerox machine, right? And also you can say it's a cell-free technique. So here you can see, okay, 95 degree. I mentioned over the 94 degree. Anyone you write over the 94 degrees Celsius stand separate. That is known as the denaturation. See, you can see the hydrogen bonds broken down and stand separate. Once the stand separates, what happens? We are going to reduce the temperature to 50 to 60 degrees Celsius. There, what happens? Primer goes and attach, right? That is known as primer annealing. Our primers bind to the template. And here, this kind of primers are known as what? Forward primers, right? And this will be known as the reverse primer, right? So where this primers goes attached to the 3' end. This is the 3' and this will be your 5' end. This is your 3' and this is your 5' end. So if this is 3', you need to write your 5' and this you need to write as 5'. 5' to 3', which is moving like this. So reverse primer, and here this is moving like this. 5' to 3' primer, forward primer. Once within 1 minute, this primer annealing will be happening, and further, when we increase the temperature to 72 degrees Celsius, which is optimum temperature for to work for your tag DNA polymerase, at this temperature, the tag DNA polymerase goes and attaches to this double strand initiation region, and that will start synthesizing the new strands of the DNA molecule. Once they synthesize the new strands of DNA molecule, we got the from two strands. Earlier we had only this blue strand, no. After this one cycle, this is the one cycle, okay? The denaturation, annealing, and the extension. Denaturation, 1 minute, annealing 1 minute, and this extension, 2 minutes, okay? This is one cycle. In one cycle, we got within how many minutes? 2, 3, and then 4. Within 4 minutes, we got 4 strands, okay? Like that, we can go for the repeating, okay? We'll go for the repeated thermal cycler. And here also you can make out, okay? Some of the, based on the PCR kit machine, sometimes the temperature may vary, but don't worry, okay? You can write either 94 degree Celsius or 98, or some of the book they'll also mention 95. Any one you mention, okay? Don't panic. Okay, at this particular temperature, we can see the denaturation temperature is increased to the temperature for the separation of the DNA strand, reduced to 55 degrees Celsius. Here you can see this is the forward primer and this is the reverse primer. Okay, temperature is decreased to allow primers to base pair to complementary DNA template. Okay, this is, you can also write this as the definition in exam. They want this one for 1 mass. Then when you from 55 degrees Celsius to when we increase temperature to 72 degrees Celsius, this tag DNA polymerase goes and attach, and the polymerase extends primer, and the polymerase extends primer to nascent DNA, and we get the new DNA strands. Okay, here you can see this is the first cycle. We had only the this particular blue, right? This is the first cycle. Okay, we had earlier only this particular blue DNA strand. After the one cycle, we got here this one. Okay, after the first cycle, we got here two blue strands and 2 red strands. So 4 copies we got here. 1, 2, 3, 4 copies. When you go to the second cycle, what happens? We got 8 copies. How 8 copies? This DNA strand again they separate. Okay, again they separate here. As and when they separate again, every time they'll go through the denaturation process. After denaturation process, primers goes and attach here. Here also primers goes and attach here. Then you are going to get the newly synthesized DNA strand. Okay, every time each all the DNA strand, newly synthesized DNA strand further will go through the repetition of the same cycle. Then you are going to get the uh, amplification of the DNA molecule. Third cycle, we got 16 copies. Fourth cycle, we got 32 copies. Then 30th cycle, you are going to get the 2 billion copies. Okay, we can see in this PCR machine, exponential amplification can be seen. Why we can see this exponential amplification? Because of the repeated thermal cycle. Okay, the region of interest is amplified exponentially in presence of the attacked DNA polymerase. Kindly pause this video and make a note of this particular flow chart. Okay, here you can make out the same flow chart I have given. Pause this particular video and make a note of this one. Okay, here you can see denaturation, annealing, and extension. This is the primer one, and this is the primer two. You must keep in mind always need to attach this primer to only to the 3' end and only to the 3' end. Okay. So here you can see this is the original template DNA strand, blue color one, red one is the new strand. This was the first copy. Okay, from this two strand we got here. After first cycle, second copies. After second cycle, we have the 4 copies. Okay, then each time this will separate. This will separate. This will separate again. From this, we'll get the 4 strands. From this again, we'll get the 4 strands. From this again, like that goes on multiplying, and ultimately we'll be able to amplify the DNA molecule into many number of copies. I hope you understood here. Okay. Let me conclude here with the definition of the PCR and with the applications. Okay, what is PCR? They ask me. The PCR is a technique that enables the multiple copies of DNA molecule which is generated by enzymatic amplification of target DNA. Right? Enzyme. This PCR amplification is possible only because of enzymes. So we can mention this as the enzymatic amplification of target DNA. It is a piece of equipment that makes the PCR mixture to be incubated in a series of temperatures that are varied from pre-programmed manner. We can give this particular definition also. So what we observed in this particular PCR, we observed the 3 mechanism, that is steps: denaturation, annealing, extension, right? Denaturation maybe 94 or 92 degrees Celsius for 1 minute. Annealing 54 to 52 to 54 minutes you can go on for 1 minute. Okay, or some of the books they'll also mention 50 to 60 degrees Celsius. Then extension 72 or 74 degrees Celsius for 2 minutes. Okay, for one cycle we need how many minutes? 4 minutes. Okay, so the machine then also tell here this one, instead of this 1 minute denaturation they may write 2 minutes, and you also 2 minutes, and you also 3 minutes or 1 minute, like that may vary, but you write this particular standard one enough for your examination. Denaturation 1 minute, annealing 1 minute, extension 1 minute. Okay. Who discovered this PCR machine, or who demonstrated Carey Mullis in the year 1985? How to study these PCR products? The amplification product is studied in various ways in order to gain information about the DNA molecule that acts as original template. Okay, a wide range of procedure have been devised for studying this particular PCR product. There are three techniques are particularly important. Okay, that is gel electrophoresis of PCR product. After the PCR, we can run that particular PCR product or sample through the gel electrophoresis to understand the particular DNA molecule. Then we can also go through to study this particular amplified DNA molecule. We can also do the cloning. Okay, again the same sample will be taken, subjected to the plasmid DNA. Again plasmid DNA will be introduced into the um, a host system, and further we can go for the understanding that particular uh, uh, introduced DNA molecule. Okay, and also to study this PCR product we can also perform a sequencing of the PCR products. Okay, sequencing, that is nothing but the understanding the arrangement of the nitrogenous bases, a study of arrangement of the nitrogenous bases. Okay, that can be done to this particular PCR product. So we can go through DNA sequencing. Okay, we have two types of DNA sequencing. One is the Sanger's method of DNA sequencing. Other one is the Maxima Gilbert method of DNA sequencing. Any one methodology we can perform. Okay, I'll repeat. DNA sequencing can be performed by two methods. One is the Sanger's dideoxy method of DNA sequencing. Other one is the Maxima Gilbert method of DNA sequencing. By using these two techniques, we can go through sequencing of the PCR products. Now let us go through the limitations. Most polymerase enzyme, however, are able to rectify errors by reversing over the mistake and resynthesizing the correct sequence with the proofreading. Okay, almost all the DNA polymerase enzyme will be having proofreading, right? Always all the DNA polymerase move from 5 prime to 3 prime, right? Always move from 5 prime to 3 prime. But whereas this particular enzyme, they are lack of proofreading. Tag DNA polymerase lack of proofreading, but most of the most of the other DNA polymerase enzyme, they have proofreading. They'll again correct by going through the 3 prime to 5 prime direction. Okay. So tag DNA polymerase appears to be lack of proofreading activity and as a result unable to correct its errors. Okay, this means that DNA synthesis by synthesized by the tag DNA polymerase is not always the accurate copy of the template molecule. Okay, the error rate has been estimated at one mistake for every 9,000 nucleotides that synthesizes. Okay, so amplified DNA should be studied directly rather than being cloned. Directly study means we can go through the electrophoresis technique or by DNA sequencing method. We can study this particular PCR products. So this is about the how to about the limitations of the PCR machine. What is the limitation? Major drawback here? This particular DNA polymerase lack of proofreading. Okay, if there are any kind of mistake, they cannot check that particular check that particular mismatched DNA that cannot rectify the errors by reversing over the mistake. Now let us go through the applications of PCR. Okay, new applications are created every day. Every day this PCR machine is updating, so there are so many applications can be seen. Okay, to amplify the DNA fragments isolated from the organisms, to propagate the DNA for gene manipulation, on construction of the DNA libraries or genomic libraries, and this PCR machine is also used in sex termination of embryo, and this is used in forensic science, that is in DNA fingerprinting. PCR products can be used for mapping genes. Then PCR products can be used as props. Props are nothing but the single standard uh, segment of DNA. Okay, used to detect genetic diseases. PCR can be used to identify genotypes. PCR can be used to sequence DNA directly. And this PCR machine is also used for the detection of viral infection, for example, COVID-19 infection by using RT PCR, then also used to detect the tuberculosis, which is caused by mycobacterium, then cancers, then prenatal diagnosis, okay, and then recombinant DNA technology for the construction of the, I told you already that genomic libraries, okay, and also to find out the thalassemia. Many more applications are going to come under this particular PCR machine, okay. Further applications, that is PCR is used extensively as a research tool which has grateful which has which is which has greatly improved our ability to study genes. Okay, and most studies in molecular genetics involves the use of PCR. For example, DNA amplified by PCR can be used for DNA sequencing as a probe in northern and southern blotting and to generate clones. Okay,
PCR amplification can be seen in medicine as well as in anthropology and archaeology also. It is also important tool in the biotechnology industries. PCR has made important contributions in many areas, which include inherited diseases, cancer research, then forensic science, prenatal diagnosis using PCR. Study of DNA polymorphism using PCR is also can be seen. Human genetics using PCR, recombination data recombination data, PCR in gene therapy, PCR analysis of plant viruses, detecting mutations, molecular mapping through PCR, preparation of molecular markers. Okay, by using PCR for research, etc. Many more applications can be seen with this particular PCR in this particular advanced era. Okay, I hope you understood this today's class PCR. I, Dr. Pratibha K, signing off for today. Stay tuned with me. I'll be back with some more videos. Thank you for watching.